Bismillahirrahmanirrahim. We have started our chapter photosynthesis. Today we will talk about action and absorption spectrum. I'm Dr. Zahra Noreen, your course instructor of Plant Physiology 1, course code BOTN 3119. First of all, absorption spectrum. How does one illustrate the efficiency of light absorption and its physiological effects? In the coming slide, a graph is shown in which the absorption of light by a pigment, in this case chlorophyll molecule, measured as relative absorbance, is plotted as a function of wavelength. The resulting graph is known as an absorption spectrum, which emphasizes the correspondence between possible excitation states of the molecule and the principal bands in the absorption spectrum. The height of the absorption curve, or the width, as represented in the figure, at any given wavelength reflects the probability by which light of that energy level will be absorbed. More importantly, an absorption spectrum is like a fingerprint of the molecule. Every light absorbing molecule has a unique absorption spectrum that is often a key to its identification. For example, there are several different variations of the green pigment's chlorophyll. The pattern of the absorption spectrum for each variation generally resembles that shown in the figure. Yet each variation of the chlorophyll differs with respect to the precise wavelength at which maximum absorbance occurs. Because light must first be absorbed in order to be effective in a physiological process, it follows that there must be a pigment that absorbs the effective light. A typical action spectrum for leaf photosynthesis is compared with the absorption spectrum of a pigment that is extracted from the leaf from the same leaf, which mainly has chlorophyll in it. The action spectrum has peaks in the blue region and in the red region, which also correspond to the principal absorption peaks of the pigment in the blue region and in the red region. So in this diagram, two actually plots are shown, two spectra are shown. The dotted red line is representing the typical action spectrum of the leaf photosynthesis, and a solid black line is representing the absorption spectrum of the pigment chlorophyll. Because we people know that chlorophyll molecule जो है उनका जो absorption है maximum वो ब्लू रीजन में होती है और रेड रीजन में होती है. सो इसका absorption spectra और इसका जो एक्शन स्पेक्ट्रम है दोनों ही एक दूसरे से कॉरेस्पॉन्ड करते हैं. मीन्स के एक्शन स्पेक्ट्रम एक्चुअली प्रोसेस का होता है, यानी कि कोई प्रोसेस जो है वो लाइट की प्रेजेंस में किस हिस्से में ज़्यादा हो रहा है वहीं पे उसकी मैक्सिमम की पीक जो है वो शो होगी. क्योंकि ये जो एक्शन स्पेक्ट्रम है ये इस टाइम पे लीफ की फोटोसिंथेसिस का है, तो फोटोसिंथेसिस किस हिस्से में ज़्यादा हो रही है लाइट की, वो है जी ब्लू रीजन और रेड रीजन. तो जो अब्जॉर्प्शन स्पेक्ट्रा है वो चेंज हो सकते हैं पिगमेंट्स के, यानी कि वो डिफरेंट हो सकते हैं फ्रॉम द एक्शन स्पेक्ट्रम. लेकिन क्योंकि जो क्लोरोफिल है उसका मेन कंट्रीब्यूशन है टुवर्ड्स फोटोसिंथेसिस, और जो बाकी पिगमेंट्स हैं, एक्सेसरी पिगमेंट्स हैं जैसे कैरोटीनॉइड्स हैं या ज़ैंथोफिल हैं या फाइकोबिलीन्स हैं, तो उनका जो काम है, उनकी जो अब्जॉर्प्शन की जो पीक्स हैं वो डिफरेंट रीजन में होती हैं अदर देन ब्लू एंड रेड, इससे मुख्तलिफ हैं. तो उनका ज़रूरी नहीं है कि उनकी पीक्स जो हैं वो एक्शन स्पेक्ट्रम ऑफ द फोटोसिंथेसिस के से कॉरेस्पॉन्ड करें. बट क्योंकि क्लोरोफिल इज द मेन पिगमेंट विच इज रिस्पॉन्सिबल फॉर फोटोसिंथेसिस, सो इसका एक्शन स्पेक्ट्रम जो है ट्रूली कॉरेस्पॉन्ड करता है विद द इसका absorption spectrum truly correspond करता है to the action spectrum of the photosynthesis. Here is another representation of the absorption spectra of the visible light, means this is the whole spectrum, and these are the absorption spectra of different pigments like chlorophyll A, B, beta carotene, lutein, phycocyanin, phycoerythrin, etc. Action spectrum. One of the first tasks facing a photobiologist when studying a light dependent response is to identify the responsible pigment. One important piece of information is called an action spectrum. An action spectrum is a graph that shows the effectiveness of the light in inducing a particular response plotted as a function of wavelength. The underlying assumption is that light most efficiently absorbed by the responsible pigment will also be most effective in driving the response. In other words, the action spectrum for a light-dependent response should closely resemble the absorption spectrum of the pigment or the pigments that absorb the effective light. A comparison of the action spectrum with the absorption spectrum of the suspected pigments can therefore provide useful clues to the identity of the pigment responsible for photosensitive processes. As an example, a typical action spectrum for photosynthesis in the green plant is shown in the previous slide. It is compared with the absorption spectrum for a leaf extract that contains primarily chlorophyll and some carotenoids. Note that the action spectrum has pronounced peaks in the red and blue regions of the spectrum, and that these action maxima corresponds to the absorption maxima for chlorophyll as well. And this is part of the evidence that identifies a role of chlorophyll in photosynthesis. Engelmann Experiment. Some of the first action spectra were made by Theodor Wilhelm Engelmann in the late 1800s. Engelmann used a prism to disperse sunlight into a rainbow that was allowed to fall on an aquatic algal filament. A population of oxygen-seeking bacteria was introduced into the system. The bacteria congregated in the regions of the filaments that evolved the most of oxygen. These were the regions illuminated by blue light and red light, which are strongly absorbed by chlorophyll. Today, action spectra can be measured in room-sized spectrographs in which a huge monochromator bathes the experimental sample in monochromatic light. But the principle of the experiment is the same as that of Engelmann's experiment. Action spectra were very important for the discovery of two distinct photosystems operating in oxygen-evolving photosynthetic organisms. Before we introduce the two systems, However, we need to describe the light gathering antennas and the energy needs of photosynthesis. Schematic diagram of the action spectrum measurements by Theodor Wilhelm Engelmann is shown here. Engelmann projected a spectrum of light onto a spiral chloroplast of the filamentous green algae, Spirogyra, and observed that oxygen-seeking means aerotactic bacteria collected in the region of the spectrum where chlorophyll pigment absorbed, means the blue light region and the red light region. The Spirogyra evolved oxygen, and the aerobacteric bacteria are gathered more in the blue light and in the red light region. This action spectrum, so this is useful in the construction of an action spectrum, which is shown here. So this action spectrum gave the first indication of the effectiveness of the light absorbed by accessory pigments in driving the photosynthesis. On the left side of the slide, here is a comparison of the absorption spectrum of different pigments and the action spectrum for photosynthesis rate in plants along with the Engelmann experiment is shown. Means this action spectrum is constructed with the help of Engelmann experiment in which the aerotactic bacteria, where they are gathered more, will make the peak of the action spectrum. And here are the absorbance peaks of different pigments, means chlorophyll A, B, and carotenoids are shown. Conclusion. So what is absorption spectrum and what is action spectrum? So different definitions are given here for conclusion, or you can say the summary. Absorption spectrum is a plot of the absorbance of a light as a function of wavelength, or it displays the amount of light energy taken up or absorbed by a molecule or substance as a function of the wavelength of the light. For example, in case of chlorophyll, absorb the different fractions of visible spectrum of radiant energy, and the percentage of each wavelength absorbed is called absorption spectrum. Action spectrum is a plot of the efficiency of the light in causing a reaction as a function of wavelength, or an action spectrum depicts the magnitude of response of biological system to light as a function of wavelength, and the relative effectiveness of different wavelength for photosynthesis is also called as action spectrum. In this diagram, the different regions of visible spectrum are shown along with the absorbance spectra of different pigments like chlorophyll A, chlorophyll B, carotene, beta carotene, phycoerythrin, and phycocyanin are shown here. You can see their peaks in different regions of visible light spectra. Now the light harvesting antennas and photochemical reaction centers. A portion of the light energy absorbed by chlorophylls and carotenoids is eventually stored as chemical energy via the formation of a chemical bond. This conversion of energy from one form to another form is a complex process that depends on cooperation between many pigment molecules and a group of electron transfer proteins. The majority of the pigments serve as an antenna complex, means they are collecting light and transferring the energy to the reaction center complex, where the oxidation and reduction reactions leading to long-term energy storage takes place. Here is the basic concept of energy transfer during photosynthesis is shown, means light energy is absorbed by many pigment molecules. These pigment molecules, after absorbing the energy, will transfer it by energy transfer to the other molecules in the antenna complex, and finally they will dispose their energy, it means light energy, into the molecule of the reaction center, where it accepts the electron from the donor like water and release the electron to another acceptor, for example the many proteins in the Z scheme or the electron transport chain. which is so this part of the transfer of energy is known as the electron transfer, and this portion is known as the energy transfer. So here many pigments are involved on not only the chlorophyll molecule but also the accessory pigments which are involved for the absorption and then transfer of the energy, which will finally poured to the reaction center. Now, what is the importance of light harvesting antennas? What happens if these antenna complexes are not present, or what is the need of these antenna complexes? How does the plant benefit from this division of labor between the antennas and the reaction center pigments? Even in bright sunlight, a chlorophyll molecule absorbs only a few photons each second. If every chlorophyll had a complete reaction center associated with it, the enzymes that make up this system would be idle most of the time, only occasionally being activated by photon absorption. However, if many pigments can send energy into a common reaction center, the energy is kept alive a large fraction of the time. In 1932, Robert Emerson and William Arnold performed a key experiment that provided the first evidence for the cooperation of many chlorophyll molecules in the energy conversion during photosynthesis. They delivered very brief flashes of light, 10 to the minus 5 seconds, to a suspension of the green alga Chlorella pyrenoidosa and measured the amount of oxygen produced. The flashes were spaced about zero point 0.1 second apart, a time that Emerson and Arnold had determined in earlier work was long enough for the enzymatic steps of the process to be completed before the arrival of the next flash. The investigators varied the energy of the flashes and found that at high energies, the oxygen production did not increase when a more intense flash was given. The photosynthetic system was actually saturated with light at this time. Here is the relationship of oxygen production to flash energy, which is the first evidence for interaction between the antenna pigments and the reaction center. At saturating energies, the maximum amount of oxygen produced is 1 molecule per 2500 chlorophyll molecule. And at that time, to evolve 1 molecule of oxygen, about 9 to 10 absorbed quanta are required to produce 1 molecule of oxygen, and about 2500 chlorophyll molecules are engaged for the production of 1 molecule of oxygen with the help of 9 to 10 absorbed quanta. Here you can see at low intensity जैसे जैसे light की energy बढ़ती है तो evolution जो है वो oxygen की that is also increased, but with the passage of time stage is arrived where higher intensity of light will not contribute for the increase in the production of oxygen. Here you can see the system has been saturated and no further change can contribute to the increase in oxygen evolution. In their experiment of measurement of the relationship of oxygen production to flash energy, Emerson and Arnold were surprised to find that under saturating conditions, only 1 molecule of oxygen was produced for each 2500 molecules of chlorophyll in the sample. We know now that several hundred pigments are associated with each reaction center and that each reaction center must operate 4 times to produce 1 molecule of oxygen. Hence, the value of 2500 chlorophylls per oxygen was calculated. The reaction center and most of the antenna complexes are integral components of the photosynthetic membrane. In eukaryotic photosynthetic organisms, these membranes are found in the chloroplast, and in photosynthetic prokaryotes, the site of the photosynthesis is the plasma membrane or the membranes derived from it. Reference books are given here to further enhance your understanding of the subject and especially the topic. This is all about today's lecture. If you have any question or query, you can ask on Google Classroom, WhatsApp group, or you can email me.